The overall goal of this procedure is to differentiate neural stem cells derived from human-induced pluripotent stem cell colonies into mixed cultures of neuronal and glial cells. This model is suitable for drug and chemical toxicity screening.
This in vitro model can be applied for assessing chemical-induced perturbations of signaling pathways that are activated during the process of neuronal and glial differentiation. The main advantage of this system is that it is a human model derived from induced pluripotent stem cells that represents alternative to traditionally used cancer cells and animal primary cultures, and allowing to study neurotoxicity in mixed population of neuronal and glial cells. The procedure will be demonstrated by Francesca Pistollato, our postdoc, and Carolina Nunes, a postgraduate student from our lab.
Begin by passaging the hiPSC colonies. Working under a stereoscopic microscope at 4x magnification in a laminar flow cabinet, use a 1 milliliter syringe with a 30-gauge needle. Cut the stem cell colonies in squares of about 200 microns by 200 microns.
Cutting out the colonies requires good manual skills to obtain fragments of equal size. The use of commercially available cutting tools can help.
Then use a 200 microliter pipette to detach the colony fragments from the dish surface by gently pipetting the medium underneath to lift the pieces. Transfer 100 or so colony fragments to a qualified matrix DMEM F12 coated 60 mm dish, filled with 4 milliliters of complete hiPSC medium.